Good afternoon, everyone. This is Lydia from BMK Gen. I'm thrilled to welcome each one of you to our latest webinar. Today, we will explore from Gen to visualization, basic and advanced drawings. Our webinar today will last for about 25 minutes, followed by a dedicated 5 minutes for your questions and answers. You can freely submit your questions by using the Q&A button at the bottom of your screen. We are looking forward to receiving your inquiries. And if any questions are left unanswered, we will contact you to reply via email. Also, we'll share the slides and the recording of this webinar via email. Last week, we discussed the process of gene filtering. Now you have a gene set based on your selected parameters, but how can you visualize the results of these genes? How can you customize images to suit your needs? Let's address these questions step by step. In this session, we will cover three main topics to help you make the most of our cloud platform's capabilities. First, one-step drawing. Discover the simplicity of creating visual representations of your gene data with just a few clicks. Second, advanced drawing tools. In the case of cluster heat map, dive deeper into our advanced drawing tools using cluster heat maps as a case study. And third, interactive filtering and plotting, COG bar chart. Explore the interactive features of our platform with the COG bar chart. But before we dive in, let me introduce our distinguished speaker for today's webinar, Johnson. Equipped with extensive expertise in bioinformatic analysis, shines in his role. Holding a master's degree from Beijing Forestry University, China, he embarked on his professional journey as a bioinformatics analyst post-graduation, specializing in high-throughput sequencing data analysis, especially Pacific Biosciences data. Johnson honed his skills with precision and dedication. His career trajectory led him to BMK Gen, where he transitioned to the Overseas Bioinformatics Department. Here, he assumed the pivotal role of Cloud Platform Project Manager, steering the team towards innovation and efficiency. With a blend of technical prowess and strategic vision, Johnson continues to make significant contributions to the field, driving advancements in bioinformatics with unwavering determination. Without any delay, let me hand over this virtual stage to our master, Johnson.
Thank you. Hello, everyone. It's Johnson. Today, we will talk about from gene to visualization, basic and advanced drawing. Time goes by. This is our third presentation today. Do you remember what we've talked by far? The first webinar, we talked about how to use BMK Cloud to get analysis report. You can select your data already on BMK Cloud or upload your own data. Submit an analysis task. I introduced to you overall content of the final report. You can get expression matrix. Maybe it is the most important result for mRNA-seq reference analysis. You can simply monitor your analysis process using BMK Cloud. You can change analysis parameters such as p-value, FDR, fold change, and so on, even after you get the final report. I think this function is very flexible. The second webinar, we talked about how to use BMK Cloud to get filtering results. Maybe you want gene set based on specific feature annotation, such as some functional pathway, or based on published paper, such as a specific gene ID, or based on feature sequence, such as a motif. You can get one or more gene sets in your needs and simply get visualization results using BMK Cloud. We talked about getting gene set in multiple criteria. Briefly, we will get target list by far. A picture is worth 1,000 words. After we got target gene set, how to get visualization results, how to customize images without coding, how to get further filtered gene list based on visualization results. We will talk about these situations using BMK Cloud this time. In order to solve these problems, we have 3 topics today. First, I will introduce one-click drawing function to you. You can get visualization results easily and quickly using this tool. But sometimes the image wouldn't suit your needs well. So we will talk about advanced drawing tools, which allows you to change font or layers settings. BMK Cloud has a very flexible function that you can do interactive filter in images. I will introduce these tools to you. Today, we will go through main functions in slide firstly, then demonstrate to you step by step. Firstly, let's talk about one-click drawing. It's easy to understand. Get your gene set, do one click, and wait for the results. Here are some notifications. The gray background means that the image is done, while the other type of the blue title means the working is still running. We must keep in mind that visualization results are based on the current gene set. In this screenshot, it means that our images are based on the 403 genes above. Maybe you can't see this picture clearly, but I will demonstrate how to observe it step by step later. You may wonder what kind of images we can get. Here is the detail. You may get 4 types of visualization results. First, you may get classification and annotation results, such as COG, KOG, GO, and KEGG classification image. Next type is visualization results for functional enrichment analysis. You may get enrichment results and the bubble diagram of GO and KEGG. For expression pattern analysis, you may get cluster heat map. And we also have interaction network results. You may get protein-protein interaction networks. So we get visualization results using one-click drawing from target gene list. There are also some functional buttons. Here, there are also some functional buttons. Image wheel will get you to a new windows where we can customize the image or do some further filter. The 2 functions are both helpful. We will talk about this later. One-click download will get a zip file which contains all the images results, while results download will download a single image for current page. There are many customized tools in the image wheel. First, we can change the font family, font size, font face, or the font color in the image. Different publications request its own style. Next, you can modify color using in your image. You can use the color we pre-prepared for you, or use your own settings. Layer settings can change boundaries, axis, legend position, or other detailed settings. You can try this part by yourself. These 3 parts are for most images. For this COG classification image, it is a bug block indeed, so you can sort the column as you need. Here, we have gone through all the functions briefly. So let's deal with some specific situations. Here are the needs. First, I want to draw images with genes related to sugar and customize the image, change the font parameters and color in image. Next, I want to do further gene filtering, such as genes related to cell wall. Notice that this gene set must inside previous gene set sugar. How to deal with these situations? I will demonstrate to you step by step. First, opening the main interface of your final report. This is the main interface of the BMK Cloud, and click the report here. Remember that this is the main interface of your final report. The first step is to get the gene set related to sugar feature annotation. We talked about this part last presentation. Do you remember how? Here, select the gene search function. Using the feature nodes, here you can see we have 28,000 genes in total, and we filter about feature nodes about sugar. And wait a minute. Here you can see we got about 400 genes in total. This is the gene set. Our first goal is getting drawing. So we do the one-click drawing. Select the page, and here you can see one-click drawing. Click it. This may take a while. On the left, you can see all the 4 images we will got. And here you can see the title of the image is still blue, which means the drawing process is still running. Here, KEGG classification turns to gray, which means this process is finished, such as this. This with gray background is finished. We use COG classification image for further analysis. Now, let's customize this image to show, and I will show you how to personalize your results in your needs. You can click the image on or the button image wheel into the customized windows. Let's change the window. Let's change this image step by step. Click this image. And you can see some functional button above. First is the text adjustment. We can change the font family, font size, font face, or the font color. For example, I want to change the font family to Times New Roman. So click here, you can find this font. So you see, all the font family turns to Times New Roman. BMK Cloud is very flexible. Think about a special situation. I want to change the title of the image here. I want to change the color of the title into red and with arrow font family. This situation hardly occurs in our daily research, but I want to show you how to deal with this situation using BMK Cloud. You can see when we in the text adjustments mode, all the text has a blue background, which means our adjust will change this part. This time I want to change this title only, so I unclick this the part I don't want to change. The axis title and the legend I don't want to change. I only want to change the title of the image. So I click this part only and want to change this into color red. So you see this, I change the color to red of the title, but the other part is still black. and I want to change the font family here. So we can see the legend and the axis title is still Times New Roman, but the title of this image is turned to arrow. After changing, click axis button here, which means text adjustment mode is finished. Then let's change the color. Here you can see this is the default color. You can change the color with the functional button here. You can select colors we prepared for you. Maybe I want to change to MPG. So you can see the color is changed. Notice that we offer the gene color collection inspired by scientific journals, such as here. AAAS refers to American Association for Advancement of Science, and NPG refers to Nature Publication Groups, or you can set your own color here. After we change the color, we click the exit, which means we finish the color adjust mode. Next is layer settings. There are many functions, and if you are not sure which to change, the default parameters are suitable for most situations. In this part, you can change layer settings, such as size of the whole image here. And you can change legend position, axis label, or data label. For example, I want to show the data labels here. You can see the number of these genes is layout, and you can, if you want the gene label. If you don't want the data labels, you can unclick here. You can click all these buttons by yourself and test what will happen when you click it. After you change exit this mode, the last part is specific for this COG classification image. You know, this is indeed a bar plot, so you can sort the columns. Click here. Maybe you want the columns from small to large, or you want the columns from big to small. This is all on your needs. Access this function. So we customized the image. You can download this image by click here, pick to download, and you can get the zip file containing this image. So we finished the second goal, customize my result. Next goal is to do the further selection based on the visualization result. We talked about this part last presentation, remember? For most images in BMK Cloud, you can do interactive filtering. We want genes related to cell wall, right? Here you can see cell wall is the M type. Cell wall is the genes with the M label. So click here. You can see here, data filtering is clickable. Click it, and you will get a gene set with 59 genes in total. This is different from the previous result. Remember, do you remember we have about 400 genes in total about sugars, and in this gene set we have nearly 60 genes in total for the sugar and the cell wall. Let's back to this result, and let's try these functions in another scatter plot using bubble diagram, for example. This is a bubble scatter plot. This is not the same of the bar plot wall. Here you can see the functional button is similar to the function above. It also has the text adjustment, the color adjustment, and the layout class settings. This is not as much as the change before. And you can click this to change the position of the axis and the other details if the italic of the label. In a personalized, you can change the function axis axis. You can change the axis. Our previous example, it is a bar plot, so you can change the columns. This is a scatter plot with different axes, so you can change the x-axis. This time is the rich factor. I want to change this to the gene ratio, so clicking it. The result will turn to the gene ratio automatically. Same to the before, you must access the edit mode and download the picture result. So let's back to our slide. Let's summarize here. Today, we talked about from gene to visualization results. We did one-click drawings and download the images. And I introduced to you about the advanced drawing tools. In a case of COG classification image, we change the text, change the color, all the layers, and the sort of the column. And I introduced to you our flexible function about interactive filtering and plot. You can click the bar into the genes list. Next presentation, we will deal with the 6 scientific topics with real data. We will get an analysis report, check parameters, and find a logical framework and insights. Hope you come and join me. That's all. Thank you. Any questions?
Thanks for Johnson's presentation. He explains the topic in great detail. I believe we all have a good understanding of from gene to visualization, basic and advanced drawings. Now we have received some questions regarding the topic, but due to time constraints, we can only answer 2 questions on stage. The rest will be answered by email. The first question is, how many times can I adjust my visualization results?
Oh, this is a good question. Maybe our customers is not sure how many times they can customize these pictures. Here, I want to tell you that you can customize or personalize your images without any limitation. You can change the image step by step, one by one, until they suit your needs well. So don't hesitate and try BMK Cloud and track BMK Cloud now.
Okay. The next question, where can I find the modified images if I forgot to download them?
Oh, this is a good question. Because when I showed you that I clicked here, download this picture. When we download this picture, we will get a zip file into our computer. And when we access this image windows, the BMK Cloud will ask you whether to save the picture to your personal data, and you have a file name. Here, you click OK, and BMK Cloud will save this image to a specific pathway, and you can save this way. You can get the image. And where is the image? Let's back to the interface of the BMK Cloud. Here is the interface of the BMK Cloud. You can check the data, and your image will listed here.
Okay, thanks for Johnson's answer. Due to time constraints, we apologize for not being able to answer our questions, but we will answer them by email after we have finished. As we conclude today's session, I want to express our gratitude for your participation. Look out for the follow-up email with the webinar summary and the promised recording, and make sure you check our social media page for future webinars. Thank you again. Have a nice day.